Next class, guess what? You guys are moving way around all over the place. So last class to sit on your butt the whole time, at least for a while. Yeah, I don't either. It's wearing me out. Hey, what's up, Tubbs? What up, Tubbs?
Hello.
How's your mama and them? Start at 2:05. We got 2:05. We got 7 minutes. Did you get to see snow the other day, Tubbs? Did it stick it where you live?
It was like raining snow, but not
Yeah.
really. It wasn't sticking.
Yeah, it didn't stick where at my house either. What can you do? Better luck next time. I got a lot of snow on New Year's Day, though. It was pretty magnificent. I got 5 inches of snow, not at my house, but where I was on New Year's morning. It was beautiful. I'll be right back. Give me 10 seconds. Precious. Met your sister yesterday. Yep. Yep. Can I get a yep? Can I get a precious? Good morning, weirdos. Here they come. Stun him out. Good morning, weirdo kids. Oh, make sure I'm recording. Oh, recording. Good morning. What's up, Daniel? Good morning, Emma. So, Emma, are you going to be here or not, baby? Emma?
I'm at home.
Yeah. Are you at home only some of it? Are you here only some of the time?
No, I wasn't feeling good in the morning, so.
Ooh. Gotcha. Gotcha. Good. All right. Well, I hope you feel better later today. We'll wait 3 more minutes to get started. Man, you want to be here on Friday if you can, because we're gonna, in class, we're gonna be getting up. Everybody else will be in breakout rooms, but you guys will be getting up and moving around the class and doing some things. So it'll actually be you won't be sitting on your butt the whole time. So if you feel well, right? Obviously, don't come up here if you don't feel well. Be like, oh man, I just really want to go to class. No. Let's calm down with that. They only let the pretty kids come first before class started. I guess all the ugly kids will be here in a minute. Oh, speaking of which, there's one now. Speak of the devil, look at all of them. And good morning, kiddos. All right, well, be quick. We start in 1 minute. Boys, yeah, I think you missed you last time. Boys, sorry, I think we missed you last time. I hope that you enjoyed the video. All right, cool. We got 1 more minute before we start, 12:04. As you guys can see, those of you guys that are here already, today is all we'll be doing is dealing with questions that you guys posed. I believe from this class, there was 17 questions that you guys posed, and we'll be dealing with those. After that, it should be a pretty sufficient review of the cell cycle. We'll move on to meiosis versus mitosis. But the student-led Q&A is all we're going to deal with today. So I'll take those 17 questions. I'll break those down over the next 50 minutes. And then I'll release you guys to take the quiz. And then if you didn't submit a question, I can show you how you can still do that. But first things first, we're going to deal with the questions that were submitted over the past 3.5 days. We'll deal with those first. Okay, it is 12:05, so let's go to the questions. When you guys submitted the questions, and if you guys want to look at the questions that are coming up, you're certainly welcome to. You were to submit the question with your name and what slide it came from. So that's what I'll do is I'll read the question, and then I'll go to the slide at which you're referencing. So it was Alan Mock. Alan, you asked the question, if certain protists divide in a way that is intermediate to binary fission and mitosis, which one is it typically classified under, or is it under a different classification? And that's a really good question. Let me go to where I'm getting. You got that question from. You said 29. And just as a review of what binary fission is, and binary fission, again, for prokaryotes versus eukaryotes, prokaryotes, all they do is simply just replicate the chromosome. We're not entirely certain how they actually separate the chromosome, but they start at the origin of replication. They go through, they copy their DNA. They have 2 copies of DNA, and then they just literally just pinch inward, have a cleavage furrow after separating the DNA, which is what we're not quite certain how they actually do it because they don't have spindles. They separate those 2 chromosomes, and then they split their cell wall and membrane cytoplasm, and they're done. 2 sister or 2 daughter cells. That's it. It's a pretty simple process. We don't know exactly how that process evolved from prokaryotes to eukaryotes. Your question is, what do we classify it as? Technically, we classify it as mitosis, as you know, there is a meiotic phase of this in diatoms, but is, you know, that's kind of just a knowledge question. I don't know if that really answers your question. Mark, is that how do you feel about that answer?
I mean, you said it was part of mitosis, then that would
Well, yeah, I'm
question.
sorry. Say again?
You said it was technically it would be classified under mitosis because there's that stage, so that would
Yeah.
satisfy my question.
Okay, yeah, I just kind of felt like that was a pretty clear question. Wasn't a whole lot to it. I mean, not that I'm criticizing your question at all. I think that's what's interesting about it. It's just the different ways they do it. In this case, like instead of the centrioles being anchored here at either pole, and then the centrioles have the spindle fibers that connect to the connect to the sister chromatids and then drag them apart. They do it inside the nucleus. So it's very similar. They're just doing it inside the nucleus, while in this case, we get rid of the nucleus and then separate it with the cell wall. Here, the spindle fibers aren't anchored to a certain point with centrioles. They're just anchored from cell membrane to cell membrane, and they just pull the actual nucleus apart. It's just different. We don't really know how those intermediate steps evolve. We just know that this is potentially how it could have occurred. Elosa, you went to 30. It says, where do the spindle fibers from? Where do these spindle form in the dinoflagellates? And they do, and they anchor here on the surface of the cell membrane. And if you remember, when I was teaching you guys Tracy, you have integral proteins, and they anchor to integral proteins, these proteins that span the distance of the cell membrane, and they anchor to those, and then they pull it apart. It does take ATP. Does that satisfy your answer?
Yeah, it does.
Okay. You guys are going to take, some of you guys are asking when we take the quiz. We take the quiz at the end of class. Unless you want to go ahead and knock yourself out and take it now, you're certainly welcome. I saw 2 people took it last night. If y'all want to take it before class, knock yourself out. I can't stop you. Let's see. If cyclin-dependent kinases, that's the third question. This is from Tracy. If cyclin-dependent kinases is an enzyme, then what is it acting as a catalyst for? And you took that from this slide. It's a catalyst for promoting the cell cycle. That's what it really is a catalyst for. It's a catalyst for a process. It does make a product. So CDK here, cyclin-dependent kinase, and this is important for your quiz today. When you look at CDK, CDK doesn't degrade. It's always present within the cell. So that's just remember that, you know, enzymes don't break down because of a reaction. CDK is going to always be present. CDK will react with cyclin to make a compound called MPF. MPF is the catalyst for mitosis. So if I really had to say, you know, because it's a cause, if I had to say what MPF or what, you know, it's a catalyst for, I would say the cell cycle. Because cyclin, there's multiple cyclins for each little step. This is a very simple version of it. So the one that you're seeing now is just the catalyst for mitosis. And maybe it will be better if I broke it down and showed you in another way. So let me show you another document real quick. This might help you understand it a little more. I put this together for you, Tarleton. Yes, ma'am. So cyclin, you guys can't see this document, correct?
Yes, sir.
Okay, beautiful. So I got cyclin, consider it this way. So cyclin-dependent kinase and cyclin itself. So if I take cyclin and cyclin-dependent kinase, then it's going to react to make a molecule called MPF. And there's going to, and again, there's genes, right, that this is going to turn or turn off. So let's say that this is going to turn this gene on. Remember that DNA doesn't just, it's not just a random molecule. There's little regions on it. There's little sections on it called genes. And so this regulator here would say, oh, okay, well, we've got MPF. We need to go ahead and go to mitosis. We need to step outside of interphase and go to mitosis to progress, in this case, to go through prophase, metaphase, anaphase, telophase, cytokinesis. You're going to separate your sister chromatids. You're going to separate the DNA into different nuclei and divide into 2 new daughter cells. After those daughter cells are created, you want to get rid of this cyclin. So this cyclin, in this case, at that point, would get broken down. You would break that cyclin down, and then cyclin-dependent kinase would persist. That molecule would continue to persist and just wait for more cyclin to be produced. That's essentially what that would be like. I don't know if that answers your question, Tarleton. Does that satisfy your question?
Yeah, it answers it.
Okay, well, let's try to look at it even another way. So think of it this way also. We'll put it in a graphical form. So if I have MPF, MPF is going to, we're in G1, we just divided, in this case, we just divided, and that cyclin is going to start breaking down. And when it breaks down, MPF is going to decrease. And if you have no cyclin at all, you're just going to stay there in G1 or GO. And the cell cycle will essentially, this blue line will stop. If I were to have MPF, cyclin start increasing again, if I had more cyclin, then I'm going to increase my MPF, and then the cell cycle would actually start to increase as well. We would get out of GO. Maybe that's even more helpful. But MPF is really going to be the control to go into mitosis. But then as soon as you get to G1, that MPF is going to fall off. You're going to get rid of the MPF because the cyclin is going to break down. Okay, let's go back. Oh, we've got a question. No, you don't. You really don't. And so, Abby, you can look in the chat. You guys really don't need to know as much as I'm explaining to you. What you really need to know is that there are internal and external signals in the cell cycle. There are going to be an internal signal, and an external signal is going to tell the cell to turn on and to go into the cell cycle. And there's going to be signals that tell it to stop as well. You just need to know it's not a random process. If it was random, we would have random growth all over our multicellular bodies, and that would be out of control. You don't really just need to know that much. You just need to know that there are external signals and internal signals as well. And this is going to be, which would this be? In this case, cyclin-dependent kinase and cyclin. Would that be internal or external? You guys drop it in the chat. And drop it in there in private. Is it internal or external? And I want at least 10 of you guys to take the risk of dropping that in there. Is cyclin and CDK?
What internal?
Is that an internal regulator for the cell cycle or an external regulator? What is that? An internal or external? Thank you, thank you. I've got 3 of you guys that have taken a shot. There's 4, 5, 6, 7. Give me 3 more. Okay. So it is going to be, and it was an even split, 5 versus 5. It's actually an internal regulator. So let me go back to the PowerPoint just to be clear about this too. So remember in this experiment, this is internal regulation. So this is a regulatory process of the cell cycle. But what we did, we put these 2 cells together, and remember, they didn't go from, in this case, G1 to S phase without merging these 2 cells. We took these 2 cells and we fused them together. And this is how we understood that there was an internal regulation for the cell cycle. We knew that there was internal regulation. External regulation is where we did this, where we had these cells, we took the cells, we cut them up, we digested the proteins off of the surface, so they basically, just remember these, in Tracy, You've got transport, you have receptors, though. The receptors are the big deal. So in the receptors, if you digest those proteins off the surface, you no longer can receive a message. And these cells didn't know to grow, even though they had everything they needed to grow. They didn't know to start growing because they had no ears to basically receive a message that says to grow. So you would receive a message to grow externally, and then you'd have an internal mechanism to actually control it once you got the message. So there's really external and internal controls. And CDK and MPF, this is all going on inside of the cell. This is regulating the cell cycle within the cell, not outside of the cell. These are internal signals that control the cell cycle, not external. Okie dokie. So let's see who the next person asked the question. This was Tabitha. Is it possible for cyclin not to be degraded? Is it possible for there to be a scenario where there wasn't enough CDK to degrade the cyclin for the next cycle? Okay, so two parts to the question. Is it possible for cyclin not to be degraded? Yes. So I'm going to ask you a question, Tabitha. And are you here, Tabitha? I don't even know yet.
I am sorry.
Okay, that's all right.
I'm not feeling well, so I decided I didn't want to put my camera on.
Don't worry, I can't see your face. Oh my God, you guys can't see the screen, can you? Shoot, why didn't you guys tell me you couldn't see that? God dang it.
I thought you were just talking.
Yeah, I know. Don't let me. And I was trying to point something out, and I was just realizing that you couldn't see what I was trying to explain, and that's no good. So I'm going to go back really quick. I want to make sure that you can see this. This is what I mean by external signals. So the external signals are here. Or this is an experiment that showed that there were external signals. The extracellular matrix, that's the cell surface of the of a cell, and it has these receptors. They digested those receptors off with an enzyme, and then they put them in two groups. With, in this case, this is a growth factor, and this was without a growth factor and with the growth factor. And when you did, even with these factors that said grow, they did not grow. They didn't. They could not hear the voice, so to speak. It would be like if I cut your ears off or I plugged your ears full of clay and you couldn't hear me tell you to do something. You wouldn't know what to do. So there's basically a signal coming from the outside that says, hey, you need to grow. And then once you do get that signal, you've got internal regulation for that growth. So there's going to be two situations here. And then I'll go back to Tabitha's question. Is there a scenario where there's just cyclin can stays? Yeah, there is. That is possible if you don't degrade the cyclin. I'm going to ask you, Tabitha, what do you think the consequence of that would be? If you did not degrade the cyclin, if it didn't break down after mitosis?
The cell would stop the cycle, or just like, I guess it could cause like it gets stuck in that phase, I guess.
It actually, what it actually would do is it would jump from, if I had enough cyclin, and you can see here, cyclin plus CDK makes MPF, you would just go from mitosis, you would jump straight to mitosis again. The cell would just divide. It would also divide without replicating the DNA, which would not work, right? Because then it would not be able to express any genes. A cell would just divide with no nuclei. It would be a non-functional cell. So you have to let that cyclin degrade, go down. You want to go through interphase, growth, synthesis of DNA, doubling your organelles, and then divide again. So it's and when you do this, you've got a full complement of DNA, because your DNA is going to be replicated in that phase. So if I just had cyclin just remain steady, your cell would just divide. I mean, to be honest with you, I don't even know what would happen after it divided once and it didn't have any DNA. I don't even know if it could have a signal. I don't know if it would keep producing cyclin. I don't know how that would actually even work after it produced it again. I guess the cyclin that's there just wouldn't degrade and it would just keep rolling. It'd just be a cell without a nucleus. That'd be really weird. Does that, the second part of your question, I need to bring your question back up again. You had the two parts to your questions. Second part of your question was, let's see, where is it?
The second part of my question
Oh,
was
yeah.
just reiterating because I wasn't sure if I had written it down clear enough because
Oh, okay.
my own question was confusing me, so I just fixed it.
Oh, you just went on to the document and changed it?
No, so the second part of the question is the same as the first part. I just reworded it because
Oh,
for
okay.
me the first part didn't make any sense.
You were just clarifying. Okay. Did I answer the question that you intended to ask?
Pardon?
Did I answer the question that you intended to ask?
You did, yes. Thank
Okay.
you.
All right. You're welcome, sweetheart. All right. So we're going to go to Mukhtar. Mukhtar, you wanted same slide, same two slides. These two. What are the two types of regulatory proteins in the cell cycle and how do they fluctuate? Well, the internal regulators, and this is really important for your quiz today. That's a great question to ask, Mukhtar. The internal regulators really are these proteins. They are going to be cyclin and CDK. I feel like we've beat this to death by now, and I'm sorry to repeat it, but I do want to be clear. You do have external regulators as well. You have these are going to be internal, and I'll use a different document. And if I don't bring it up, say something. Oh, there's a couple of chats. Let me see what you guys are saying. Oh, I'm not sharing your screen. Yeah, I'm sharing it now. Okay, let me go to another document to maybe make this a little more clear. So let's say that these are two tissues. This is a tissue, and this is also going to be a tissue. We got two tissues here. These little blue dots are going to be density-dependent inhibition factors. Now, what does it mean to inhibit something? Let me ask somebody. So, Precious, what does it mean to inhibit something? I'm sorry? I can't hear you. To what? To end it. Speak, yell at me. She's in the room with me. Oh, she's going to put it in the chat box. To take in, no, to inhibit is to, can somebody else help me? What does it mean to inhibit something? If I inhibit you from breathing. What?
Inhibit, isn't it like it's something to like stop you from
Prevent.
doing it?
Yeah,
Prevent.
stop, prevent you from
Hold you
breathing.
to prevent it.
Yes. Love y'all. Okay. Yeah, it's to stop it or to prevent it, right? So these are going to be factors that tell these cells, hey, you've got a neighbor next door, stop dividing. They're all sending these little signals out, these little proteins, but sharing them between each other. And another way to maybe think about this would be, think of it this way. So I've got, this is a cell, it's a terrible looking cell, and this is its receptor. And this cell is its next door neighbor. And it also has a receptor. And then it's got this, I can't change color. I think this is, yeah, it's the only color I've got as an option. So now this signal is being sent from internally and released and binding with this and say, hey man, I'm next door. There's no need to keep growing. You've got a next door neighbor. And this cell's doing the same thing. It's binding here and say, okay, we're next to each other. Remember, cells can't talk, so they have to have some way to pass signals back and forth. But if I were to come here and wipe these cells out, just like if I skinned my knee, in this case, this would be like Roethlisberger getting smoked and having problems and skinning his knee and hurting himself. Pittsburgh Steelers really choked on Sunday. So now you've got all these cells missing. Well, they can't hear each other anymore. Like these little blue dots, they can't get quite back and forth. They can't really make that jump. That's too far. So these cells now know, hey, well, you're going to divide. Let's go through mitosis. Let's fill in the gap. There's nobody here. Think of it also kind of like this is East Coast and this is West Coast. And this is like manifest destiny, right? We got to fill in the gap, man. We got to fill this gap in. This is vacant land. It wasn't vacant. It was held by Native Americans, but this is like the gap between the two, and you filled in that area. There was nobody there. In this case, they didn't think there was or care that there was anybody there. There weren't people, there weren't cells like them. And so they just copied it. And then once they fill in that gap, well, let's get some of these, let's clone some of these signals. We drop those signals over here. Everybody starts hearing each other again. And they stop dividing. That's what we want. In an idealized world, that's what we want. Oh, the blue dots are going to be inhibition factors. They're little proteins. Like this is what these are. These little blue dots are these inhibition factors. They're just little proteins that they're passing back and forth, these external signals that say, hey, you need to grow or not grow. And that's what these are. They're just little proteins that they synthesize and they drop out. They use exocytosis to secrete, and then they have a receptor that binds to it. This little, I don't know if my mouse can work here. This little, I don't know what color this is. I think it's brown. Gets passed back and forth, and they know to stop. Here are the little blue dots. Does that answer your question, Abby?
Oh, yes, thank you.
Welcome, sweetheart. Okay, so let's go to the next contestant on The Kids Are Right. You guys ask great questions. You guys didn't ask as many questions this first period, but I do think that you guys asked some really good questions. That was Mukhtar. Thank you for asking that one, Mukhtar. That was really getting down to the point. Let's go to Earl. Earl is on slide 37. Slide 37. It's here. Sorry, it takes a minute for me to jump from one place to the next. And your question, Earl, is what are some examples of internal and external signals? Jesus, okay. Does that help? Did I just answer your question, Earl? Are you even here, Earl?
Wait, what? What was happening?
What? No, I'm just, you asked the question. Your next question was, what are some examples of internal and external signals? So I feel like I've already gone over those. Could you give me an example of an internal signal?
Oh, the cycling thing.
All right. What about an external signal? Could you give me an example?
No, I forgot.
Oh, Jesus. Somebody help him. What could one be?
The cycling?
That was internal. That's internal. Cycling's internal. What about this one right here?
PDGF?
Yeah, a growth factor, some sort of growth factor. So the one I just showed you, the one I just showed you before was going to be a proximity dependent inhibition factor to stop you from growing. So this would be an example of an inhibition factor. If their cells are all next to one another, they're communicating. They know, stop dividing, quit crowding me out. If I scrape some of them off, they'll fill in that gap. That would be one of those factors that I was trying to explain to you. These density dependent inhibition factors. I mean, they're not stopping dividing for no reason. They recognize that one another are there. Think of it this way, Earl. You're pretty comfortable with me. I feel like. I mean, you castigate me and stuff and say crazy stuff on Remind all the time. So I'm gonna. So,
because I beat your score?
Because what? Yeah, because you beat my score. Shut up. Lopez did that. Now, just pretend for a moment that you and I are in class and everybody could see this. God, I can't do it in here. I wish somebody was in here. But I wish that we didn't have this pandemic. If you were in here and you were standing in the middle of the room and I walked up to you and I just kept walking, would you eventually put out your arm and say stop?
Yeah, I guess.
Yeah, you wouldn't want me to walk until my nose touched your nose, right? We'd look like we were giving each other hugs and kisses. That's not going to be okay. Now, I felt like you're pretty comfortable with me. You'd probably let me get pretty close. But there's other kids in here that would probably, if I was got walking toward it, and like I'm looking at Precious, Precious is like, Jesus, man, if you were walking towards me, I'd throw something at you before you got within 5 feet of me. Is that right, Precious? Yes or no? She said yes. So there's those external factors, like it's that, like don't get in my personal space. That's what's going on here with these cells, the same way. Some cells are a little more comfortable and get really tightly packed, and we looked at that when we saw tight junctions, like in your intestines or your heart, where these cells have to be very close together so there's not leakage between them. Other cells, not so much. They can be a little more spread out. Have I satisfied your question, Earl? Yay or nay?
Yay.
Yay. All right. And I know you cheated on that quiz too. So,
Yeah, sure.
So hush. Okay. All right. So the next question, and then no one put a name with this one, but I was able to find it. I don't remember who it was, but I was able to find the name because I had to go literally look and see who changed the document. And so you did get a grade. Is what causes the cell to have a copying error in the first place? And that's a mutation. There's a lot of reasons for that. I don't remember who asked that question. There's a lot of reasons.
Me.
It was, who's me? Is that Liao?
Yeah.
Okay, there's a lot of reasons. And smoking could be one. So like cigarette smoke causes mutations. UV light causes mutations. Sometimes there's, yeah, it could either cause a delay or it could cause a cell cycle to go out of control. It kind of depends. So we're going to learn about that in probably about 2 weeks. Actually, it's about 2.5 weeks away before we start learning about mutations. But there's a lot of causes for that. Many causes. Sometimes it's just literally you make you're writing something down, like some of you guys copy each other's homework, and it's a little different, right? But other times it's going to be because of pollution. It could be UV light. Those are probably the two biggies. And then there's other things called telomere decay, which I'm not even going to try to explain in here right now. So I'm going to jump to another one because we're going to learn about that later. Emma? Oh, there's a question in the chat box. Okay. Let's see. What does it mean when cancer cells exhibit neither density dependent nor anchorage dependence? Let's take a look at that. What does it mean? So I've got another document for you because that was a good question, Emma. I like some of you guys' questions were really solid. So I've got a little drawing for you too. I was thinking when I was looking at these, I was like, how can I explain this? It's going to be a good way to explain it. So, okay, so let's look at anchorage dependent first. So imagine an anchor like a boat. If I am a boat and I'm drifting around, I throw out an anchor so that I'm grounded, so I'm stuck to something, so I'm fixed to something, so that I am adhered to something. I am going to be grounded. So that's anchorage dependence, and that's what cells need. This is in order to grow. They need a substrate to be standing on or fixed on or kind of, in a lack of a better word, glued to. What happens is when a cell becomes dislodged, it doesn't grow anymore. It's just sitting out here, not anchored to anything, and it doesn't grow anymore, or at least it shouldn't. Occasionally, though, there are cells that doesn't work that way, and so they do start to grow. And when they start growing uncontrollably and they're floating around, they land in a sensitive area and they start to grow a tumor. And that would be an example of cancer. But these no longer are exhibiting anchorage dependence. They shouldn't do that. You don't want this to happen, because if this does happen, it could kill you. So you want to get rid of this. You want to kill these cells. This is what it's normal. This is what you want to happen. You want these cells to have a surface that they're growing on. When they become anchorage, they're not dependent on anchorage. That's a problem. That would be or that would be cancer, or at least a tumor. Again, ligands is a general term for a cell signal. Density dependence or proximity dependence, that's where you're just passing the message back and forth saying, I'm here, I'm here, I'm here, I'm here, I'm here. Now what happens is some cancers, instead of having a receptor that looks this way, they go through mutation where the receptor is shaped a different way. And they don't hear that message. So that receptor doesn't match the shape of this ligand. So while this cell does, I don't know if I can get my mouse to work, hold on. While this receptor does work and it tells the signal, don't grow, somebody's next door to you, this receptor does not receive this message. It's there was a mutation that changed the shape of this protein. And so this cell will then decide, well, I'm gonna divide, I'm gonna grow. I'm going to go out of control, and this cell could become a tumor. It could start dividing uncontrollably. Now, does that satisfy your question, Emma?
Yeah.
You're not Emma. Hush. I'm talking to Emma.
Oh, sorry. I thought you said my name.
Emma, are you still here, sweetheart? Well, I'm assuming that you went to the restroom. Okay, Tracy asked the question, how frequently can cancer cells divide? It depends, sweetheart. It really depends. It depends on the cancer. Some are aggressive cancers and some are not. I'm not an oncologist and I couldn't exactly tell you which cancers are more aggressive than others. It really does depend on the cancer. And a classic example of a cancer that's a very slow cancer would be prostate cancer. So males have an organ called a prostate, and that prostate, when you do get cancer in your prostate, that's something that takes a very long time for that tumor to grow. They're a very slow growing cancer, and that's not one that you should generally be too worried about. If you catch it early, your survival rate is almost 100%. Other cancers are going to be different. A friend of mine has a father, a really good friend of mine has a father that has a brain cancer, and it's killing him. It's a very aggressive cancer. He's been able to slow it down, but it's something that's given him a very hard time right now. And Tabitha is bringing up that brain, sinus, and lung cancers are generally tend to be very aggressive cancers. And that's true. And lung cancers are several different varieties of lung cancer. And lung cancers, some are more aggressive than others. My sister had a type of cancer that was extremely aggressive and it grew very quickly, but it was also very treatable. And so sometimes it's a double-edged sword. Some of your aggressive cancers are actually very treatable cancers. And then some of your less aggressive cancers are not very treatable, but they're slower growing, so you have a long survival time.
Are they more treatable because people focus with them more because the more aggressive ones are more common?
You know, I don't know. I'm not an oncologist. I couldn't tell you. I would start lying to you. And that's the one thing I will tell you for sure is that I'm just not going to lie. I don't really know, but I do know. But that's an idea that makes sense. I mean, there's a lot of logic in your theory that may be the reason why, but I don't really know. Let's see. Slide number 42. This is also from Tracy. Tracy, what happened to you? You decided you're just going to ask a bunch of questions?
You said first ask questions.
Yeah, so you did. I think you asked like 4. I was like,
That's
what?
3.
Oh, is it just 3? This was 42. So you said on 42, what makes a cancer malignant? Is that what you were saying?
Yeah.
Okay, so let's use the breast. This is a classic breast cancer. And for you ladies, and I'll kind of preface this for a moment. So this is obviously your breast, but you guys can start giving yourself self-examinations at your age. Your breasts probably are still, some of you guys are still growing, are still growing. And some of you guys may have already stopped growing your breasts. So you, about the time that they're, I'll say about the while they're still growing, they still might feel a little hard and lumpy. You get some tissue in there that's still going through the growth process, and they may feel kind of tough. This granular
Mr.
tissue.
Pope,
Oh,
you're
you
not
can't see
scared.
it.
You're
Oh,
not
you
sharing
can't
your
see it.
screen. Yeah.
Thank you, thank you, thank you. They're like, what? This is your breast? This is a bunch of people's faces. So this granular tissue, sorry, here's your breast. This granular tissue here can feel kind of lumpy at your age. It can start to smooth out later on in life. Nevertheless, when you start to give yourself a breast examination, because you can feel these tumors growing in your breast, because breasts tend to be pretty soft, you can feel little lumps there. When you do feel one, you need to go get what they call a mammogram, which is really where they just scan your breast and look for density of cells. And then if they find a really high density of cells in an area that they're not supposed to be in, then they know that they need to do a biopsy. And when a biopsy is where they take a sample of those cells and they look and see is it cancer or not. Now, your question was about it being malignant. So malignant is where it goes from one tissue to another tissue. So in this case, this is going to be this granular tissue that goes to these milk ducts, and then it gets secreted out of the nipple. So let's say that this tumor is just on these milk ducts, these ducts where milk would flow to feed an infant. If it spreads to neighboring tissues, now it's in the granular tissues and it's in these ducts, then it is now what we call metastatic. It is now malignant and moving into other tissues. And this is what's dangerous. And now if it stays within the breast, that in itself isn't going to kill you. You could have your breast removed, you could go through reconstructive surgery, and have your breast rebuilt by a plastic surgeon, or just live without them altogether. So there's many females that do this. And then but what could happen is that tissue, now you have some of these cells that are not anchorage dependent, they get into your bloodstream or they get into your lymphatic system and they start moving around your body. When they move around your body, they could get lodged in other tissues. So in this case, it can move from your breast to, maybe it can move to your liver or your ovaries or another part of your body that could kill you. You need to move around? Yeah, just sign up and go. Sorry, somebody need to go to the restroom. It could go to your brain. It could go any number of places. And so that's what we talk about when we have like metastatic tumors that leave one region of the body and travel to another region of the body. But malignancy is where you're spreading from one tissue to a neighboring tissue. You're going from one type of tissue to another type of tissue. They're spreading in a way. Is maybe a better way or a more easy way to say that. Does that answer your question, Tracy? Okay. Let's see. What determines whether or not a cancer is benign or malignant? We just talked about that. Okay. That paired those right next to each other. What determines the speed or intensity of cell replication, and can it lead to some other cancer if excessive? Yeah, I mean, I don't know what exactly would determine how intense cancers could be. I can give you an examples of why our body doesn't deal with these very well sometimes. So I'm going to switch to that other document. Excuse me for a minute. There was a question from one of you guys. That was you just saying, sharing my screen. Come on, and next time and just say, hey, you're not sharing your screen. It's easier for me to hear and see it. All right, here we go. Notice I'm not talking. Rambling about something that you can't see. All right, so you do have cells, and let's say this is going to be a cancerous cell. I'm going to draw this as a cancerous cell. Remember, you have cells that are self and non-self, and you have cells that have these little glycoproteins on them. And glycoproteins are these proteins that are on the surface of the cell that are like markers. And so I'll kind of even draw out here to the side. Let's call this a marker, kind of like if I have a name tag. I have a name tag and it says, this, I am Mr. Pope and I belong in room 207 at Summit International Preparatory. So you have this marker that signifies this is a self cell. This is like me. I am precious and don't hurt me. You have another cell called a natural killer cell. It's a natural killer cell at NKC, and it's an immunological cell. And all it does is move around your body, and it's looking, I'll draw an eye here, all it's doing is looking for self and non-self cells. So it doesn't have an eye, so to speak. This is just kind of me signifying that what it does is it looks, it's going over here and saying, hey, what is this? What is this? Oh, the marker is saying that this is the correct cell. But what happens if that changes? If I have a mutation and this marker changes, and instead I get something that it looks like this, I get a marker out here that looks like this, it goes through a mutation, then a natural killer cell will attack it and kill it. It will come in here, and it basically what it does is it eats it. It will send out poisons to it. It'll send out sort of some toxins to break it down. It will try to digest it. It will kill this cell. This happens to every one of us every week, at least. I don't want to say every day, but you have cells that go rogue, that could potentially become cancer, and that your immune system takes care of all the time. So let's pretend for a moment. Let's say I have 1,000 natural killer cells for every one of these. So you've basically 1,000 versus 1. Who's going to win a fight? 1,000 versus 1. Of course, the natural killer cells win. What happens is if you get old, if you smoke, if you get overweight, your BMI is really high. If you get all those things, this number decreases. In this case, now this number becomes 10, and this becomes 500, so to speak. So your BMI could hurt this, because now you're made of more cells. Your BMI, remember, is your body mass index. You get more of a mass because you've got more cells. Your immune system doesn't grow, but the rest of your body does. So you get more mutations, and your immune system is not very healthy, and so it can't keep up with it. You smoke, and so that suppresses your immune system. It also is going to increase mutations, so that hurts you. And then, of course, you're getting old. And as you get old, your immune system weakens, and you have the more opportunity for mutations because your cells have been dividing for so long over your entire life. All these could allow that to, this situation to occur. And let's see, whose question was that? That was, there's no name on that one. No name on that particular one. I don't know, whomever asked that question, does that answer your question? Okie doke. Awesome that you are at the restroom as well. The next one is, does the apparatus control the movement of the chromosome after mitosis? Does the apparatus? What are you talking about, Stein? You didn't put a slide number on it. Stein, that was your question. I don't know what slide you're talking about. It says, does the apparatus control the movement of the chromosome after mitosis? So I'm not sure what you mean. You there, Stein? Hello? Stein. Okay, I'm going to go to the next question. Do cancer cells divide faster than other body cells? Yes, they do. And I think that's it. That was the last question. Is there any other question that I missed that you guys can see? Is there another question that somebody would like to bring up? Because the next thing I'm going to do is release you guys to the quiz. This is your chance to ask. Okay. I'm going to release you guys to the quiz. Let me show you where to find it. So what you'll do is actually, hold on, for this class, you're going to go to class content, and you go to class nope, that's Friday's lesson, and you go to class content. You're going to see the last quiz that you don't want to mess with. You've already taken that one. You're going to see a new one that says Cell Cycle Quiz 2. Cell Cycle Quiz 2. That's where you're going to find the quiz. Now, let's say that you did not ask a question, and you want to ask a question now. If you do, you can, and you can recover those points because you say I put a 50 in if you did not. However, there's one caveat. If you do ask a question to get that 100, it cannot be the same question anybody else has asked. It cannot be the same question. So you need to be very careful and look at what other questions have been asked. So in this case, I'm looking here and I can see that like Alan asked about the protus dividing, Alosa asked about the dinoflagellates, whatever, you have to be very careful and make sure that you are not asking the same question somebody else asked about. So you can recover those points. Right now, it's not open for you guys to use. I locked it to only viewers this morning. So if you didn't post a question, you didn't get a chance to ask one this morning. I'm making it editor now. You guys can ask a question, but now what you want to do is you want to go and you want to take the quiz. You're going to have 13 minutes to take this quiz. So go ahead and click it. I'll be quiet and I'll let you guys take it.
Mr. Pope,
Yep.
we could still ask questions, right? Or add questions to the, okay.
Yes. Please take your time when you're taking the quiz. Look at the command term. If it says explain, that means you need to use 3 sentences to do this. No bullet points. Do not just throw a blurb in there. They'll see the word limit. You should probably be approaching the word limit to be successfully getting all the marks that you can get. So if it's 3 marks or 4 marks, you don't want to write 10 words. That's not going to cut it. You might be lucky to get 1 point out of 10 words. I don't know. Is that what you submitted? Yeah, I submitted it. Okay. Yeah, you got a grade for you. Good job. No, you do not have any homework. If you're, you are able to leave after you've taken it, yes. You guys be good. I wish you guys asked more questions in this one.
Mr. Pope, it said
Yep.
that my test isn't saving.
It's because it hates you. Let me see if I can see what's going on. Tabitha hair. Did you answer everything?
No, I'm working on the last question now.
Okay. Just continue and see if it works. If not, I can submit it for you. I'll see if it works. Oh. Okay. Tough. If it's not letting you finish, it should not continue the time, but some of you guys are saying that it is for getting paused. If it pauses, it should not cause you to lose time. And I can submit it for you. Some of you guys saying you cannot submit it. I can submit it for you. Yes, you guys can split. I hope you guys have a nice lunch.
Enjoy your day, Mr. Pope.
All right, you guys play in the street tonight.
No, I think I'm
Yes.
gonna take a nap.
Play
Enjoy
in
your.
the Oh, we'll all try. All right, I'm gonna leave you guys be. You guys be good. I will have these graded today.